Well, thank you, Christine, for introducing me. As Christine mentioned, my name is Amin Mohammadifar, and I'm here today on behalf of Research Group for Food Production Engineering, where we believe that our mission is to design and develop sustainable food processing system and food products. Well, the main object of my presentation today is to show and emphasize how novel food processing characteristics could be aimed at improving food quality and also facilitate innovation that is needed for increasing added value. Based on what we have learned today and based on reports, we know that food processing industries are one of the most water-intensive industries. We also know that food processing industries have the highest contribution to emission of organic water pollutants. And also, we know that food processing industries are responsible for around 28% of the total energy consumption in food sector, and they are responsible for around 4 to 5% of CO2 production, CO2 emission during food production from farm to fork. Apart from these major challenges, we also know that food industries are interested in increasing yield and also quality of food products and shelf life of food products. And in parallel, customers also or consumers also interested to have safe and nutritious food and of course better sensory properties and of course shelf life. Green food processing could be considered as more or less a new concept that can meet all these expectations and challenges without the use of solvent, chemicals, and also excessive energy and excessive water. These emerging processing technologies are traditionally used to fulfill all the expectations that we have from conventional processing methods, like sterilization, pasteurization, and so on, or let's say in microbial inactivation or enzymatic inactivation. If you have a quick look at the literature in the last 10 years, you will note that we should expand our expectation. We believe that these techniques or tools, these techniques or processing methods are powerful tools that could be used for tailoring a structure and development of novel techno-functional properties. In today's presentation, I'm planning to share some of our experience and success stories in Research Group for Food Production Engineering, but I would like to point out two points. That first I would like to express my appreciation to my colleagues in Research Group for Food Production Engineering for providing some more information for me to make my picture completed. And then secondly, my focus is on mostly on electric-based technologies. So let's move on to the first processing, which is ohmic heating. In ohmic heating, basically food is considered as resistance. We apply electric field or voltage gradient. We have electric current. And based on the chemical composition of food and electric conductivity of food, a part of this electric energy is converted to heat. So we can use this technique for heating up food. And there are some unique aspects for this technology. It's very fast. We can increase heat very fast, which is great. We can get all the benefits that we usually get from HTST processes. So we can kill microorganisms, but the nutrient will be intact. And the second thing is this technique is used. So this technique is not rely on conduction. So it's like a volumetric flow rate of heating, and then all the a different part of the food sample will be heat up at the same time. So when the surface is hot, the geometric center is also hot. So this is an example of the application of this technology. We used it for pasteurization of apple juice. And as you can see, when you use ohmic heating, denoted with OH, we were able to successfully inactivated polyphenol oxidase, which is responsible for color change in fruit juice. And then at the same time, I hope it works. It's not working. Anyway, at the same time, it's not working. We noticed that the minimum destruction of vitamin C and carotenoids also belongs to the time that when you used ohmic heating or ohmic heating in combination with ultrasonic. So this was an example of traditional use of ohmic heating, which gives us interesting results and fast methods, and then we had better performance, and it was an efficient process compared to conventional heat treatment. But we moved forward. We wanted to get benefited from, we wanted to change the structural properties of food ingredients. So we used only electric field. We didn't heat up sample. We just used electric field, and we implement this. We apply this electric field to plant-based protein and animal-based protein, casein and protein that are extracted from sunflower cake. And then I just summarized results here without any graph or table. But as you can see here, by just using optimizing the electric field, we were able to change a secondary structure of the protein, and then consequently, we changed functional properties of this protein. We were able to lower surface tension and interfacial tension by the use of electric field treated protein. We changed glass transition. Glass transition moved to higher temperatures, which is important also. And then apparently we have been able to change the aggregation pathways. So we ended up with a small particle size, which is important when you wanted to make concentrated protein-based drink. You wanted to add more dry matter, but you still wanted to have fluidity. This is a big challenge now. I know that there are some projects between KU and Arla about this. And then we use this electric treated samples to stabilize emulsion, and then we ended up with high stability. So that means the hydrophobicity has been changed also, which is completely, we expected to have this because of the opening the structure. And then we ended up with making some edible films with electric treated samples and then controlled samples. You can see these are SEM, these are cross-section of these edible films, and then you can see the structure is completely uniform when you use this electric field treated samples compared to the control. And then we measure the mechanical properties of these films. We had a higher structural strength, and also at the same time we had more stretchability, which are desired quality attributes for edible films. Then we moved ahead. We tried to use electric field and heat treatment to change mineral balance in milk. It is important, you know, when you increase temperature in like when you have a sterilization or pasteurization, you are forcing calcium to go to the micelle, and then all the functional properties of micelle will be disappeared. If you wanted to have a good emulsification, it's impossible then. Because when you remove calcium, micelle will be disintegrated. Proteins are available to act in the interface. So this is important. We wanted to increase temperature, but still have some soluble calcium. And as you can see here, down there on the left, right-hand side, you can see we have 72% soluble calcium by using ohmic heat treatment. And compared to control, which is 37%. And remember, temperature is 85 in both cases. This is important when you wanted to make cheese powder because you need to have a stable emulsions before spray drying feed. and then you need to have good functionality for protein. And you know that cheese powder is made of cheese melt, which is not soluble. So this is helping us to solve this challenge. Now they use additives, melting salt, some companies to solve this problem. So we replace additive with processing. And then we just moved ahead, and then we used ohmic heating to treat milk that is going to be used for making acidified gels like yogurt. And then, as you can see here in this graph, in the green box, we have some ohmic heat treated samples that has higher structural strength and then lower synersis. Usually, when you can reach this by adding just dry matter, which like 7% dry matter to milk you have to add so that you have a strong gel. But we did this without adding dry matter. But sometimes when you increase elasticity of these gels, the result is shrinkage, and then you have water repelling of water, which is called weighing off. But as you can see here, it seems that we have been able to adjust elastic and viscose components in the structure, microstructure, in a way that we have a stronger structure, but it's still not repelling of water. So that means we have thickened that soluble part. So this is also a proof that electric field is able to change mineral balance and functionality of proteins and aggregation pathways. The next step is to use high electric field. We also use high electric field when you use high electric field when you subject gases, different type of gases, air, nitrogen, oxygen, different or argon to high electric field. Basically, you are creating a wide range of reactive species like ozone, H2O2, ions, radicals. And then you can use this complex mixture to kill bacteria for surface decontamination, for instance. But we use this technology or this reactive species to inactivate enzymes. In a project, you know that wheat germ is very nutritious, but unfortunately we have lipolytic enzyme in wheat germ, which makes its shelf life so limited. So we used this cold plasma to inactivate these enzymes, lipolytic enzymes, two of them like lipase and lipoxygenase. As you can see, we have a significant reduction on activation of this enzyme, but the thing is, we notice that reaction of different enzyme or sensitivity of different enzymes to this treatment is different. Yes. Then in the next step, we use this technology to change protein functionality. We just finished a review on this, and we are working on this aspect now. And then pulse electric field. Pulse electric field is similar to the previous one. We use high electric field, but this time food is subjected to the high electric field. And then the consequence of this treatment is that if you have cells inside your food, like bacteria or other cells, you make some pores in the membrane, and then you change mass transfer, and this way you kill bacteria. So it's good for safety reason. But you can also use this for other purposes. You can adjust the pore size with this technology so that you can extract ingredients inside the cell. I mean, you can use it in downstream for fermentation, for instance. So when you create something, you can extract those ingredients selectively. So imagine that you just break the structure of the cell, and then you just extract everything. So you need to put a lot of, basically you are putting a lot of pressure in downstream for purification. This way, you can just go for the right, components that you are interested in. We use this technology, my colleagues in our research group, they use this for extraction of protein from insects. And as you can see here, by implementing this technology, pulse electric, we were able to increase extraction yield by 13%, which is a great achievement. Well, this was just a few examples about the use of novel processing technique, not only for traditional uses like sterilization, pasteurization, but also for changing functionality, improving yield, and so on. So we know that we are at the beginning of a long way, but I think if this type of presentations gives enough motivation to governmental organizations, industries, fund agencies, and if they just move, if they just start moving in this road, it's easy. I mean, in near future, we will hear more about these technologies, and then we can implement them. I mean, most of these technologies now are used in industries, but there are some limitations that we have to solve. I hope that I hope this presentation is also in line with this. Motivation of people to move in this direction, and then move toward a sustainable production system. So that's all for me. Yeah. Sorry.